Hello friends, in this video we will be discussing the remaining questions of DU Biochemistry 2021 question paper. So let's begin. Match the names of the scientists in list 1 with their achievement in list 2 and choose the correct answers given below. So list 1 outlines the name of the scientists and we have to correctly match with their discoveries. First one is Watson and Crick. In 1953, Watson and Crick proposed the 3D double helical structures of DNA. So A has to go with the third. Now Robert W. Hawley, he won the Nobel Prize in Medicine in 1968 for describing the structure of tRNA for alanine, and thereby he linked the DNA with the protein synthesis. Actually, he shared Nobel Prize with Hargobind Khurana and Marshall Niederberg, who contributed for the cracking the genetic code mystery. So B has to go with the fourth, that Robert W. Hawley has actually proposed the Clover model of tRNA, while Marshall and Niederberg, they are basically involved with the deciphering the genetic codon. Now, Monod and Jacob are famous for their work on E. coli lacopron, while John Jeffrey is better known for developing techniques for genetic fingerprinting and DNA profiling. So D has to go with the fifth and E has to go with the first. So if we find out, then option B is the correct answer. The blood volume decreased when a mammal was bled rapidly. However, the cardiovascular changes resulting from hemorrhage could be maintained by the following compensatory mechanism: increased cerebral flow, reduction of baroreceptor activity and stimulation of chemoreceptor, reabsorption of tissue fluid in the blood, or increased release of enkephalin and beta endorphin. So we have to find out the correct answer from the options given below. Basically what happens when a blood loss is high during hemorrhage, arterial pressure decreases and cardiac output is also decreases, which is resulted in a series of compensatory cardiovascular responses to restore normal functioning. If suppose ये blood loss जो हुआ है, if this is moderate, then basically the baroreceptor signaling takes place. But if there is a heavy blood loss, then baroreceptor signaling is diminished and chemoreceptor reflexes is being taken over. So B statement is correct. Now, decreased cardiac output led to the profound fall of blood pressure and heart rate. So the pressure in the capillaries also fall down and less amount of fluid is transduced from the tissue spaces. So reabsorption of tissue fluid in the blood takes place. So C statement is also correct. Now, due to the decreased cardiac output, the cerebral flow is being decreased rather than increased. So statement A is incorrect. Now, because of the diminution of the cerebral blood flow, we often observe fainting or unconsciousness. Now, as we know that the endorphin and enkephalin, these are the natural painkillers and mostly released by the brain to the blood. So definitely if the cerebral blood flow is reduced, then definitely their amount in the blood cannot get increased. So statement D is incorrect. Now, neurohormonal compensatory changes also involve the release of renin from the kidney and ultimately angiotensin II and aldosterone increases. Both of these hormones basically ensure the more retention of sodium and water from the urinary output, which further allow the reabsorption of tissue fluid in the blood. So only B and C statement is the correct, while rest of the statements are incorrect. Cellular membranes are self-sealing in nature. If they are punctured or disrupted mechanically, they quickly and automatically reseal. What properties of such sealing are responsible for this feature? Is it due to the hydrophobic effect of membrane lipids? Is it due to the hydrophilic effects of membrane lipids? Because of the charge to charge interaction among lipids, or due to the protein lipid interactions? See, in case of cellular membrane is punctured or broken, then hydrophobic fatty acid chains of the plasma membrane, it tries to aggregate, and it is happening because of the strong hydrophobic interaction which is present in the aqueous medium. Remember, the hydrophobic interaction seems to be maximum when they are placed in the watery medium or aqueous medium. Hence, when due to the puncture, these hydrophobic groups are separated in aqueous environment, they prefer to reseal. So, option A is the correct answer. From the given intermediates in the fat metabolism, correct intermediate series is. So there are 4 options are given and we have to arrange the intermediate as per they happen during the beta oxidation of free fatty acid. So let's start. If you see it in the diagram, once we start with the fatty acyl CoA, after an oxidation step, it is being converted into the transenoyl CoA, having a double bond. Now, across this double bond, there is an addition of water molecule takes place. As per the Setzer rule, the OH is being added at the beta position, which after one round of oxidation is being converted into a keto group and the compound will be ketoacyl CoA. Now, a thiolysis steps actually cleave this molecule at the beta position and led to the release of an acetyl CoA. And now, a free fatty acid is formed, which is a 2-carbon short from the beginning. This molecule is again ready for the next round of beta oxidation and it keeps on happening in a spiral case manner till the last acetyl CoA is left. So, the order of intermediate would be fatty acyl CoA led to the formation of transenoyl CoA having a double bond, and now it led to the formation of beta hydroxyacyl CoA after the addition of the water, and last the ketoacyl CoA, which is being degraded through the thiolysis step into the acetyl CoA. So option A is the correct answer. Given below are the 2 statements. One is labeled as assertion and other is labeled as region. Assertion is bile juice helps in the digestion of food in small intestine. Reason is that bile juice contains sodium salts. So in the light of our statement, choose the correct option from the options given below. Both A and R are true and R is the correct explanation of A. Both A and R are true, but R is not the correct explanation of A. A is true, but R is false. A is false, but R is true. As we know that the bile salts helps in the lipid digestion in the small intestine. Bile juice has the bile salts which actually break down the large fat globules into small particles, which is also known as emulsification of fat. Now, due to the emulsification of the fat, pancreatic lipase can act over it. And the main component of a bile juice are the bile salts, which are composed of the sodium salts of glycine and taurine. So both assertion is correct that bile juice helps in the digestion of food, especially fat in the small intestine, and reason is also correct that bile juice contains sodium salts of glycine and taurine. So option A is the correct answer. All the membrane proteins are known as G proteins because they have a subunit that activates adenylate cyclase, hydrolyze GTP, are gated ion channel, or mediate the effects of insulin on cells. That's very easy. All the G protein known today contains a canonical GTPase fold which can bind and hydrolyze GTP. So option B is the correct answer. Which of the following technique is used to obtain the 3D structure of large proteins at atomic resolution? X-ray crystallography, NMR spectroscopy, electron microscopy, or circular dichroism? X-ray crystallography has been used in past to resolve the 3D structure of biomolecules including proteins and DNA structure. Who can forget the contribution made by Kendrew in order to determine the first crystal structure of proteins of myoglobin? The underlying principle of using X-ray crystallography is that the crystalline atom causes a beam of X-ray diffraction into many specific directions. By measuring the angle and intensity of these diffraction patterns, a crystallographer can predict a 3D model of the electron density map within a crystal, which can later be used to determine the 3D structure of a protein. So, option A is the correct answer. Rifampicin is an inhibitor of DNA replication, DNA gyrase, translation, or transcription. Rifampicin is a known inhibitor of RNA polymerase. When upon binding, it actually halts the transcription process. Interestingly, this antibiotic actually inhibit the prokaryotes because it's a specific inhibitor of prokaryotic RNA polymerase and do not influence the eukaryotic RNA polymerase. So option D is the correct answer. HIV infected patient exhibit preferential decrease in CD4 cell count, CD8 cell count, CD20 cell count or CD40 cell count. HIV invade T helper immune cells, which is also known as CD4 positive T cells, and once upon infection it causes the decline of T helper cells. If it falls below a critical level, then our body becomes susceptible towards the opportunistic infection. So basically HIV infect patients exhibit preferential decline in CD4 or T helper cell counts. In a biochemical reaction, the thermodynamic parameter that reaches a minimum is, that's again very easy, enthalpy, entropy, free energy, enthalpy and entropy both. As we know that delta G is an important indicator of spontaneity and extent of any chemical reaction. In a spontaneous change, any reaction proceeds towards those directions which led to the minimum delta G or the negative sign of delta G. Hence, option C would be the correct answer. Which one of the following radioisotopes does not emit beta rays? Carbon, hydrogen, phosphorus or iodine? As we know that C14, tritium, phosphorus 32, they all are the beta emitter, while iodine 125 is an example of gamma emitter. So option D is the correct answer. Molarity of a solution is the number of, that's very easy. A molarity is a concentration unit which defines as a number of moles of a solute in a liter of a solution. So option B is the correct answer. Ribulose 5-phosphate epimerase is involved in glycolysis, TCA cycle, pentose phosphate pathway or fatty acid. L-ribulose 5-phosphate 4-epimerase, it catalyzes a interconversion of ribulose 5-phosphate and xylulose 5-phosphate in the oxidative phase of the pentose phosphate pathway or also known as hexose monophosphate shunt, that is C. Which of the following is a non-membranous structure? Golgi bodies, microbodies, inclusion bodies or glyoxysomes? Inclusion bodies are the tiny particles that freely suspended and floating within a cytoplasmic matrix, while rest are the example of membranous organelles. So option C is the correct answer. Proteolytic enzymes synthesized as large inactive precursors are known as, again one of the most simple questions. So as you are seeing that MSc Biochemistry questions are not so difficult, which you cannot crack. So please trust yourself and start appearing. Now let's get back to the question. Proteolytic enzymes are synthesized as large inactive precursor, which is also known as proenzyme or zymogen. For example, pepsinogen is an zymogen which remain inactive due to the presence of 44 amino acid long masking peptide. Upon removing this peptide, it becomes an active form of pepsin. So proteolytic enzymes synthesized as large inactive precursors are known as proenzyme or zymogen. B cells acquire immune competence in thymus, bone marrow, liver or spleen. Development of a B cell begins in the bone marrow and in sequential manner they transform from pro B cell to immature B cells. Now immature B cells once formed, they are having an expression of BCR, which is a cell surface receptor. The cell surface receptor is once surfaced, then we can say that B cells becomes immunocompetent. So B cell actually acquire immune competence in bone marrow. Absorbance of a DNA solution can be measured using fluorimeter, spectrophotometer, thermometer or radiometer. The absorbance of a DNA sample is measured at 260 nanometer on a spectrophotometer using UV transparent cuvettes. Insulin is secreted by liver, muscle, gallbladder or pancreas. Insulin hormone that regulate the level of sugar in the blood is being produced by the beta cells of the isolate of Langerhans, which is present in the pancreas. So option D is the correct answer. The enzyme that hydrolyzes starch into the maltose is glucoamylase, alpha amylase, beta amylase, or glucotransferase. Beta amylase catalyzes the hydrolysis of starch into the maltose by hydrolysis of alpha 1,4 glycosidic bond. So option C is the correct answer. Prolactin is a peptide, steroid, nucleic acid, or vitamin. Prolactin is a peptide-based hormone produced in the pituitary gland. So option A is the correct answer. RNA-dependent DNA polymerases are known as transcriptases, primases, recombinases, or reverse transcriptases. So any DNA polymerases which are using RNA as a template in order to synthesize the DNA, they are called as reverse transcriptases. So reverse transcriptases are RNA-dependent DNA polymerases that make the cDNA from RNA template. This kind of enzymes was first discovered by the Baltimore in HIV, and for which he was awarded Nobel Prize. Statins, a popular class of drug, is known to inhibit HMG-CoA oxidase, GADPH, HMG-CoA reductase, or pyruvate decarboxylase. Statin actually blocks an enzyme called HMG-CoA reductase that is involved in the synthesis of an steroid intermediate called mevalonate. So option C is the correct answer. Which of the following scientists was responsible for the first database in bioinformatics? Max Perutz, Margaret Dayhoff, Linus Pauling, or Dorothy Rothschild? Margaret Bailey Dayhoff was a pioneer in the field of bioinformatics. She is credited for the creation of protein and nucleic acid databases, and she published first her work in the name of Atlas of Protein Sequences and Structure. Eventually, her data became the first online database. So, option B is correct answer, that Margaret Bailey Dayhoff was the first American lady which was responsible for the creation of first database in the field of bioinformatics. Specific functional group in an amino acid undergoes specific chemical reaction producing a typical color that helps in the detection of the relevant amino acid. Match the amino acid in list 1 to their specific test with list 2. So list 1 outlines the name of the test, and we have to correctly match that which amino acid can be characterized by using these tests. So let's start. Hopkins-Collet test. Hopkins-Collet test is used to find out the indole ring. Upon reaction, indole ring gives the violet color. Now, indole ring is present in which amino acid? That is tryptophan. So A has to be connected with second, that is tryptophan. Polys test. Polys test is used for the quantitative detection of tyrosine and histidine. So obviously by using Polys test we cannot differentiate among tyrosine and histidine. But in order to differentiate, we can use Millen's test, because Millen's test is very specific for tyrosine. So D, that is Millen test, can be used for tyrosine, and indirectly Poly test can be used for the histidine. That means B is connected with third. Leftover nitroprusside, that can be used to characterize the cysteine at alkaline pH. So A will be connected with 2, B will be connected with 3, C will be connected with 4, while D will be connected with first, that is option A. The citric acid cycle in respiration yields. So if we look at the diagram, we can find out that one round of citric acid cycle or Krebs cycle, the output product would be 3 NADH, 1 FADH2, 1 GTP, or 2 CO2. So option A is the correct answer. Which of the following property is not exhibited by an allostatic enzyme? So इसमें से 4 properties में से we have to find out which is not a characteristic properties of an allostatic enzyme. Sigmoidal substrate binding curve. Definitely, sigmoidal substrate binding curve is an hallmark feature of an allosteric enzyme. Allosteric enzymes की क्या-क्या properties होती हैं? They are multimeric in nature. That means they are quaternary structure. They have a typical substrate binding curve. That means sigmoidal substrate binding curve, unlike of a typical hyperbolic curve shown by the Michaelis-Menten enzyme. Then they have a cooperativity, and now they have two different sites. One is a regulatory site and one is substrate binding site. So one thing is very clear as far as allosteric enzyme, that no allosteric enzyme can be a typical Michaelis-Menten enzyme. So option B is the correct answer, that adherence to Henry-Michaelis-Menten equation is not a characteristic of allosteric enzyme. In fact, while explaining the kinetics, they have assumed that no enzyme should be an allosteric enzyme if they have to be solved by using kinetics. So option B is the correct answer. In the enzyme catalyzed biosynthetic pathway shown below, what will be the effect on the substrate A, B, C and D if the enzyme E2 is inactivated? That's very simple. As we can see, if E2 is deficient, then downstream product C and D will not form, but B can still form because E1 is still active. So option C is correct, that B will be produced but not the C or D. Select the true statement regarding nitric oxide, NO. NO is a vasoconstrictor. NO activates adenylate cyclase. NO activates guanylate cyclase, or NO stimulates platelet aggregation. Nitric oxide or NO is basically a vasodilator which inhibits platelet aggregation. So option A and D cannot be correct statement regarding NO. NO basically activate the guanylate cyclase activity, which actually inhibit the adenylate cyclase activity. So option C is the correct answer. A certain small molecule inhibits an enzyme by binding only to the enzyme substrate complex, or in other words, by binding only the substrate binds the enzyme. This particular inhibition is a typical of which class of inhibition? First of all, you should know what are the three types of inhibition: competitive, uncompetitive, non-competitive, and what is their pattern. Competitive inhibition कब होता है? Competitive inhibition is there when a inhibitor compete for the active sites. So that cannot be the answer here. Non-competitive inhibition कब होता है? When a substrate binding is being impaired due to the binding of inhibitor at some distant site. So this can also not be the answer because substrate is binding here. Now, in the case of uncompetitive inhibition, that one of the hallmark feature of uncompetitive inhibition is that it only gets activated when a substrate binds to the enzyme. That means the target of an uncompetitive inhibitor is the ES complex. So here the option D is the correct answer. Which of the following protein is not synthesized as zymogen or proproteins? Pepsin, lysozyme, collagen, trypsin. Lysozyme is an enzyme which is released directly in functional state, while pepsin, collagen and trypsin are released as pepsinogen, procollagen and trypsinogen, which are the inactive format of all these three proteins. So option B is the correct answer. A drug which prevents uric acid synthesis by inhibiting the enzyme xanthine oxidase is aspirin, allopurinol, colchicine or probenecid. Allopurinol, which is also known as xyloprene, actually it inhibits the enzyme xanthine oxidase, which actually synthesizes uric acid from hypoxanthine in purine metabolism. So option B is the correct answer. Heme containing proteins that function as an electron carrier in oxidative phosphorylation and photosynthesis are hemoglobin, catalases, chlorophyll or cytochromes. Cytochromes are the heme proteins that are involved in electron transport chain, that is oxidative phosphorylation and photosynthesis. So option D is the correct answer. Arrange the following steps of protein synthesis in the order in which they occur within a cell. DNA unzip in the nucleus, tRNA brings in proper amino acid from the cytoplasm, mRNA attaches to the ribosome cell, first codon is read, mRNA nucleotide transcribes the complementary DNA message, mRNA leaves the nucleus and goes to the ribosome, and F, a second tRNA, brings in the new amino acid. So we have to choose the correct answer from the options given below as per their sequence. So if you see it in the diagram, what is the first step is going to happen? DNA is going to unzip for the transcription process. What is the next process? Then the mRNA nucleotide transcribes the complementary DNA message, which is being done by the RNA polymerase, which uses the DNA template and synthesizes the mRNA. Now what is the next step? mRNA is going to leave the nucleus and going to cytoplasm and look for the ribosomes. Now what is the next step? Now this mRNA is going to attach to the ribosomes and first codon is read. What is the next step? tRNA brings in the proper amino acid from the cytoplasm, which is generally used to be the first codon is always AUG and generally the methionine is being attached at the N terminal. What is the last step? A second tRNA brings in the new amino acid as per the second codons. So here if you see, if you arrange in a systematic way, then A will be followed by D, will be followed by E, will be followed by C, then B, and then last would be the F, which is an option C. Which of the following statement is true about enzymes? Enzyme increases the equilibrium constant of the reaction they catalyze? No. Enzyme do not alter the overall change in the free energy for a reaction? Yes. Enzyme do not influence the rate constant of the forward reaction they catalyze? No. Enzymes speed up the reaction by increasing the activation energy? No. They actually decrease the activation energy. Basically, we should remember what is do regarding the enzyme, what is don't regarding the enzyme. Actually, enzyme fasten up the reaction and actually they influence the rate constant of the forward reaction. Second, what they do, they actually lower down the activation energy in order to fasten up the reaction. They never disturb the equilibrium constant, they never disturb the free energy change, they never disturb the enthalpy, or they also never disturb the entropy of a reaction. So here the option B is the correct answer, that enzyme do not alter the overall change in the free energy for a reaction. Arrange the following landmark achievement in a chronological order. Crystal structure of DNA, draft sequence of human genome, sequence of insulin, crystal structure of myoglobin. So we have to choose again the correct order in the chronology as they happen. If we know that insulin structure was first proposed by Frederick Sanger in 1951, for he was awarded Nobel Prize. In 1952, Rosalind Franklin took a detailed X-ray image of bDNA, using which Watson and Crick proposed the DNA structure in 1953. In 1957, John Cantu became the first person to resolve the crystal structure of the myoglobin, which was the first protein structure resolved by the X-ray crystallography. Human Genome Project was basically completed by 2001 to 2002. So the chronological order would be C, which was a sequence of insulin, A, then the crystal of DNA, crystal structure of the DNA, sequence of insulin 1951, crystal structure of DNA 1952, and followed by Watson-Crick proposal 1953. Then next would be crystal structure of the myoglobin in 1957, and then draft sequence of human genome, that is around 2001. So option A is the correct answer. A Ramachandran plot represents the sterically allowed conformation of a polypeptide backbone, gives the frequency of occurrence of amino acid in alpha helical and beta sheet structure, predicts alpha helical and beta sheet conformation of a peptides and proteins, or shows the X-ray diffraction pattern of a protein. Ramachandran plot is a 2D plot between the torsional angle phi and psi across the peptide backbone. Basically, this plot is used to visualize sterically hindered or energetically allowed region of a polypeptide. So, a Ramachandran plot basically represents the sterically allowed conformation of a polypeptide backbone. Which of the following statement is true? RNAs can be treated with urea to produce a random coil. If one oxidizes random coil RNAs in urea, it quickly regains its enzymatic activity. If one removes the urea and oxidizes RNA slowly, it will renature and regain its enzyme activity. Although renatured RNAs has enzyme activity, it can be readily distinguished from naive or native RNAs. Basically, this question is talking about the Anfinsen experiment. What he did? Actually, he took a native RNA and given the treatment of urea and beta-mercaptanthenol. Once this treatment is being given, definitely RNase has lost all its activity. Once they dialyze the urea and beta-mercaptanthenol, that means they have removed urea and actually removed beta-mercaptophenol, we found that the RNase actually regain its all activity, almost more than 95% of its activity. So basically, what he concluded that a denatured form actually gets folded because of the presence of its amino acid sequence, which was the landmark statement as far as protein folding is concerned. So basically in this, the option C is the correct answer, that if one removes the urea and oxidizes RNase, that means remove the beta-mercaptophenol, then RNase will renature and regain its enzymatic activity. Which of the following statement about the salvage pathway for the synthesis of purine nucleotide is correct? Increased PRPP level in the cell inactivate HGPRT. Decreased PRPP level in cell lead to the activation of HGPRT. Increased PRPP level in the cell led to the activation of HGPRT, or purine nucleotide competitively inhibits HGPRT. HGPRT basically catalyzes salvage pathway of purine bases using PRPP as a substrate. This PRPP dependent reaction by HGPRT actually gets increased as the concentration of PRPP increases, while deficiency of this enzyme resulted in the accumulation of its substrate, that is hypoxanthine and guanine and even PRPP. So here if we see, the increased PRPP level in the cells led to the increase of the HGPRT, that is option C. The absorption of glucose in the digestive tract, that's very simple. Actually, the absorption of the glucose is being done in the small intestine by the absorptive cells through the secondary active transporters. So here the option A is the correct answer. Which one is the heaviest particulate component of the cell? Nucleus, mitochondria, cytoplasm, or Golgi apparatus? Basically, we have to find out which is the most dense organelle present in the cell. So as we know that nucleus is the heaviest particulate component of the cell, having diameter of around 4 to 6 micrometer. So option A is the correct answer. Transketolase activity is affected in biotin deficiency, ascorbic acid deficiency, PABA deficiency, or thymine deficiency. Transketolase is basically a thymine pyrophosphate dependent enzyme, and its activity is being affected by the thymine deficiency. There are two more enzymes whose activity is also get affected by the thymine deficiency. Those are pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase of the Krebs cycle. Chymotrypsin is specific for the peptide bonds containing. Chymotrypsin is basically a typical serine protease which preferentially cleave a peptide bond in which the N-terminal amino acid to the bond is a large and uncharged aromatic amino acid, which could be phenylalanine, tryptophan, tyrosine, or even leucine. So here the option A is the correct answer. All the following statements about phenylketonuria are correct except. So basically what happens, the person who are suffering from phenylketonuria actually has a deficiency of phenylalanine hydroxylase. This enzyme is basically required for the synthesis of tyrosine from phenylalanine. So here the option A is correct, that phenylalanine cannot be converted into tyrosine in the case of phenylketonuria. Now since phenylalanine is found in all protein rich foods, hence the person who is suffering from phenylketonuria must permanently follow a low protein or phenylalanine diet. That means option C is also correct. Now the patient can be diagnosed for the disease by measuring urinary metabolites like phenylalanine, phenyllactate, phenylacetate, phenylpyruvate, or even phenylethylamine. So hence option B is also correct. So that means in the case of this question, option D is the correct answer, that this statement option D, it leads to the decreased synthesis of thyroid hormone, catecholamine and melanin, is not a characteristic of phenylketonuria. The most of the UV absorption of proteins above 240 nanometer is due to their content of, that's very simple. Commonly, the optical absorption of protein is measured at 280 nanometer. At this wavelength, the absorption of protein is mainly due to the aromatic amino acid like tryptophan, tyrosine, and to some extent cysteine. So here in the given question, the option A is the correct answer. One international unit, that is IU of vitamin D, is defined as the biological activity of 0.025 microgram of cholecalciferol, 0.025 microgram of 7-dehydrocholecalciferol, 0.025 microgram of ergosterol, or 0.025 microgram of ergocalciferol. If you see it in the figure, the mass equivalent of 1 IU for the various types of vitamins are given. So 1 IU of vitamin D is the biological equivalent of 0.025 microgram cholecalciferol, which is option A. The vitamins having the highest daily requirement among the following is thiamine, riboflavin, pyridoxine, or ascorbic acid. Among different vitamins, vitamin C can be taken in without having dietary complication. As far as males are concerned, they can take up to 75 mg, while females are required much more ascorbic acid, that is around 90 mg. So option D is the correct answer. Isoenzymes are chemically, immunologically, and electrophoretically different forms of an enzyme. Different forms of an enzyme similar in all properties, catalyzing different reactions, or having the same quaternary structure like the enzymes. Isoenzymes are those enzymes which catalyze the same reactions but occur in a number of different forms. All these different forms are different in terms of either chemically or immunologically or even electrophoretically migrations. A deficiency of folate led to beriberi, megaloblastic anemia, pernicious anemia, or hypochromic microcystic anemia. Low levels of folic acid can cause megaloblastic anemia, in which the RBC precursors are way larger than the normal cells in the bone marrow. So option B is correct. With this, we sum up our video. If you like our content, please do subscribe in large number and comment, like and share. Thank you very much.